So, good morning, ladies and gentlemen. Hello, everybody. Welcome in Innsbruck. On behalf of the organizational team, I would like to welcome you here in Innsbruck to Bioblast 2022, those who are here in person and also those who join via the live stream. So this is also something new for us. Before we start with the exciting scientific program, I have to share some general announcement with you. So, first of all, I need the other slides. A second. I need to start this presentation with the mobile phone, the general announcement we had. So sorry, taking time away from this first speaker. So, yes. First of all, very important, please switch off your mobile phones or switch them to the airplane mode. If you require internet connection, there are the passwords, Bioblast 2022, hang up on different places. Then the second very important thing is that, can I moderate those slides? Because they are actually turning all the time and I cannot talk as fast. Sorry.
It's not possible. We have to go. It's programmed. We have to go.
Okay, it seems that the slides are turning faster than I can talk. A mistake in the organization. I'm sorry for that. Yes. Then, second of all, at 12:30 we have a group picture outside next to the registration desk. So on the stairs we queue up. Paolo will take a picture, please show up there right after the session, so that you are also on the photo. Then we take timekeeping very seriously. So we have usually 2 chairs. Right now there is only one. The other one is delayed. They have to be very strict in timing. They also have a bell, as well as you have a monitor in front of you showing you the time. Showing you the time, and when your time is almost over, it turns orange, and when it's definitely over, it's red. For those who stick in time, we have a small present, but only those who stick in time. Yes. Then my next announcement is that you saw in the program that Nick Lane is with us. He recently published his new book. So we also have some copies of the book here, which you can buy at the registration desk for 25 euros. Please notice that you can only pay in cash. So good that I have some notes, because I would definitely be confused now. So we also have an Early Career Investigator Award foreseen for the best poster at the conference. We have a committee of 3 people. This is Elettra Leo from Italy, Tuli Cambre from Latvia, I think, sorry, and Dominic Besta. The committee will walk through the posters in the poster session time and ask the presenters for short presentations on their posters. So please make sure to be there. Outside, we also have some artwork from Odra Noel. So please also use the coffee breaks to walk through the artwork. There are also some sculptures from Lois Arwin-Dalfaret. Look at the posters, enjoy a coffee, walk to the exhibitors and get updates on their latest products. For the social program tonight, we would recommend wearing a mask in public transport. For those who don't have a mask with you, we have them and we can hand them out. Hiking shoes are necessary if we hike up. We have to see what the weather forecast is. It's not that bad at the moment, but if it's raining completely, then we have an alternative to bring you up there by car. And yes, yeah, and if you definitely need to go there up by car because your physical activity doesn't allow it or whatever, please contact me beforehand so that we can arrange that. And if you have any questions during the conference, please contact me. I would also like to refer you to Carolina, but unfortunately she's sick. She sends her greetings and hopes that you enjoy the time here. But unfortunately she cannot be with us. And then, before handing over to Erich, there is 2 more comments. First of all, we have a live stream, so please be aware of the cameras here with walking around. Secondly, if you want to ask a question in the discussion, then just raise your hand, don't get up. And the last very important information is that your name badge is also the entry ticket for all the social activities we have scheduled during the conference. Now, I wish you a nice time. An exciting time. I would like to thank all the supporters and sponsors, and I would like to hand over to Erich now for the welcome words. Thank you.
I want to continue with the welcome. Sorry for the technical problems that Verena faced. This was just a different channel for presentation. So this will be running over in breaks, but now I should have control over these slides. So welcome in Innsbruck to Bioblast 22. With this one, I heard the mouse. Okay. I would like to start with thanks. So usually one has these slides at the end. I start with this and thank Luisa, Lisa, and Paolo particularly, but not only those, but a whole team behind this celebration, Bioblast 22. For us is a celebration. Thank you, first of all, to join us for this celebration. For us, it is 30 years Ouroboros. We started 30 years ago, and there is many more members in the team. I just select a few to thank. So why Bioblast? Bioblasts are the taxonomic unit of bioenergetics. So we started the journal Bioenergetics Communications. Bioblasts are what we are studying. Mitochondria, aerobic bacteria, plastids, chloroplasts. So this goes back to Richard Altman, and in 2010, we had a mitochondrial physiology conference where Nick Lane and I totally disagreed about the bioblasts, and we continued to disagree and share our disagreement. So Richard Altman, as Nick published in his 2005 book, Power, Sex, Suicide, Mitochondria, and the Meaning of Life, there Nick pointed out that the osmium staining was a very important aspect of Richard Altman's book. And since Nick gave me the original Altman, 1894, I read it in detail. It's in German. And the osmium staining, as all the mitochondriacs know, engaged in electron microscopy, that is a staining for bacterial, mitochondrial, plastid membranes. And Altman viewed the protoplasm as a colony of bioblasts. In the 2005 Mitochondrial Physiology Conference, I referred to this. Well, mitochondria are not just organelles, they are organisms in our cells. Whether they have lost 99% or 98.5% of their genes to the nucleus, well, still they are organisms in our cells. They are the aliens. I quote here, microorganisms and granula, well, these are now recognized as the mitochondria, are at an equivalent level and represent elementary organisms. Thus, we want to describe them by the common term bioblasts. So here you see pancreas, here you see actually symbiotic bacteria, the granula, the symbiotic mitochondria. And the endosymbiotic theories, there is not one, there is about 20, I guess, endosymbiotic theories, but they all link the mitochondria and the plastids to their free living ancestors. Together, these are the bioblasts in spotlight, in the focus of bioenergetics communications. So we study bioblasts. We inaugurate now 2 years delay, BEC, because BEC was started 2 years ago, and we go now for the concept of living communications in different ways, because we had virtual conferences, they were not really living, now we are back to life. So why 22? Well, not in 2020. 22, originally we should have had the European Bioenergetics Conference here, not in this room, but in a much larger hall with 600 to 800 participants. It was delayed due to COVID-19, and in that year, 2020-21, our team was about like this. Maybe tomorrow we are like this again. So here we are, the Bioblasts are back in real life, but can we plan ahead? Yes, we try. So in 2 years, August 2024, we will have EBEC in this very building, and we expect 600, 700, if you all come, 900 participants. In next year, we decided very recently, rather before the meeting than after meeting, to think of a next one, because after meeting we are all exhausted and we say never again. So we decided before to get going with our mitochondrial physiology summer schools. It will be a summer school in end of July, and a mitochondrial physiology conference with a topic, bioenergetic communications on mitobesity. So save the date. Now, 2 years ago, BEC was actually started very much due to COVID-19. Well, there was time to do something else than traveling and so on. So we started within our next-gen O2K project, the new journal, Bioenergetics Communications, and the first paper there was co-authored by 666 colleagues, and you have it probably received already quite some time, maybe on paper even, but you have another copy now. 2 years ago already was then the number 2, so there were only 2 publications in Bioenergetics Communications, and well, it looks like a long time ago, 2020, but it was really going online on 30th of December. In 2020, the Blue Book, the 5th edition, since the first edition, 2007, this was a summer school in Schurken. We see some familiar faces, maybe here is Steve Hand. So it has a tradition, and the Blue Book is also part now of the BEC publications. In 2005 already, we started the 10+5. So this will be the mission now for you, to stick to the 10+5, with one exception, me and Nick Lane. So we introduced this with the promise, if you stick to the 10 minutes, not to the 15 minutes, to the 10 minutes, you will get a gift. So that was the gifts. They were very popular because during the whole conference it was raining. And so this gift was very useful. We should have thought maybe of umbrellas now as a gift, but we are hopeful that the weather will be okay. Okay, stick to the 10+5. We have another tradition. So in MIP 2014, Mitochondrial Physiology Conference 2014, Odra Noel presented Mitchell's Dream, and in remembering and honoring Peter Mitchell. Now, this year, at Bioblast 22, Odra Noel's focus is on the Krebs cycle that sits at the heart of metabolism. A quote from Nick Lane's website, maybe even from the book, from the introduction. And this links mitochondrial pathways to respiratory control. Odra Noel's Krebs cycle Ouroboros is at the cover of our book of abstracts of our program, and you see here the Krebs cycle backwards, I guess. And you see here Nick Lane at the Royal Institution of London presenting Transformer, the deep chemistry of life and death. And as Verena announced, we have, she did not say this, we have books here for a special discount and signed by Nick yesterday. So here I presented at the beginning, thanks to the team. I showed some slides of the teams or some faces. I don't show a slide now of Odra because she is here and she will come to the stage. So we are honored by your presence. We are honored to have on the exhibition beautiful MIP art, and you give us an introduction, and I'm always excited to have Odra with us.
Thank you
Please.
very much. Thank you. Good morning. It's very difficult to follow up that introduction, so I will just go straight into a very little introduction of how important it is to mix science with art, how much it can give to both, but especially scientists are going to gain a lot by putting a lot of art. I do have a small presentation. Thank you. So this is an unusual conference from the point of view of the big interchange, but maybe after my 10 minutes or less, you will see the advantage of why it's a very good idea to mix them. So my name is Odra Noel, it's an artistic name. I am also a scientist by background, but my role today is introduction of art. And what you are going to see here is that scientific art is a very wide concept. It is? Exactly. So the key is that you can use art to represent scientific concepts. They don't need to be only cells. They could be organelles, they could be tissues, they could be concepts, metabolic concepts, formulas. They could be very trendy viruses that are in everybody's mind. So the idea is not what you do. The idea is to put a different spin on that concept. And when they ask me to explain why you do what you do, it's very difficult to give just one sentence. So I use this word clouds. So the first one is the emotional part. You use it to inspire, to make people say, whoa, to energize, to explain to your mother that what you do in the lab is really beautiful. The second one is to pass information. And this is the one that you will be more commonly familiar with, because you all have seen some magnificent graphs or some medical illustrations who really made you click or made you even understand further. The fact that you have to put your information in a visual way makes you understand what your results may mean to other people. So the information part is the one that is more commonly used. The third one is actually the process part, which anything that you do that has some visual impact will be much easier to be memorized. So it's the recollection part of art. So just three small elements to put you into the frame of what scientific art is trying to do. First, it's always out of date, because information comes all the time, and half of the things that I have done in the past are already wrong. So you can tell that it is wrong, or you can be more kind and say, well, this is the representation of the historical knowledge at the time. And we now see a lot of the beautiful things from Leonardo da Vinci and company that are very wrong, but still beautiful, and tell us how the scientific minds of the time were thinking. So maybe in a century or two, they will look at us and say, oh, they were so wrong, but it's so beautiful. That's good too. The second part is that the colors are completely subjective. So they are all invented. And tonight, maybe when we go for a drink, you tell me, how do you see mitochondria in your mind? Because it's always very interesting to see which colors people imagine in their own research, what they do. And the third one is that they are full of information, these paintings, but you don't need to understand to enjoy it. So I have a very good friend who thinks that my liver cirrhosis is aboriginal art. I'm happy with that. If you enjoy it and it makes you think of science, that's already mission accomplished. Now you will enjoy more the more you understand what the scientist is trying to do. So I'm assuming that you are all with me with the emotion part of art and with the information part of art. So I'm going to give you an example of the memory part. And I'm going to use oxaloacetate that I'm guessing is a good friend of most people in this room. So an ox is a bull without balls in English, so the ones that you use to pull the carts. And acetate is the material that is commonly used to make all the sunglasses and the spectacles. So our oxaloacetate is going to be a bull with sunglasses. Just follow me for a little bit. Okay. So then we have the formula. We start with the 4 carbons. Where will you put the 4 carbons? It's obvious. There are only 4 places for 4 carbons. Okay. Now you have to put the carboxylic groups. The ones in the extremes are going to be easy. And then you have another one in the alpha carbon. Let's put it in the glass in there. So now we have a pyrite oxaloacetate. We need to complement, of course, the hydrogens in the spaces. So if you now see that, it's very easy to remember what the atoms were. And this is the exercise tonight. So going back to that drink, you ask to each other if you can remember, and you can write without thinking the formula of the oxaloacetate. But do this before the drinks. The colors of the mitochondria after the drinks, the formula before the drinks. And then we can write it like this or like that. So going back, now you can see that you can use these symbols and these mnemotechnics to put in a circle to memorize which are the 8 main elements of the simple Krebs cycle. You can put batteries with energies produced. You can have CO2 leaving. You have water coming in and out. It's an exercise that, because it's so visual, is far more likely. And as you know, the Krebs cycle is always going to be in the exam. So this is just a call to use the art for other ideas, not just for the typical one. And of course, this is what art has been doing all the time. So this is an example from the 16th century, but art through the ages has the same idea, especially the religious art, which is the one that is coming through us through the centuries. So you have a very clear message. This is what happens if you misbehave. You put it in a very impactful way, and people will remember the message. So this is the very simple formula. Now you just maybe have to reflect that into your own art. So have a look again. What is the message that I want to put in there? Can I put any emotion, colors, videos, sounds, impactful images, even normal, boring images, but just put a little bit that will appeal to the emotional part of the domain, and they will remember your data. So enjoy the conference.
Okay, well, I am very visually limited in comparison to that, as you'll see in a moment. But as Erich said, I've been writing a book on the Krebs cycle, and I think most biochemists are both fascinated and appalled by the Krebs cycle, because it's kind of just sits there right at the heart of biochemistry. It seems to be well, it just seems to be a lot of trivial rearrangements of carbon and hydrogen and oxygen, or at least that's how it seemed to me when I was studying biochemistry as a degree. And it came as quite a shock to me years later. I had no idea, and I don't know how many people in this room do anyway. I mean, the reverse Krebs cycle is doing literally the opposite. It is taking hydrogen and CO2 and making organic molecules. How is it that this is not taught in all the textbooks? Because it's not. And it makes so much more sense of things. But it also makes everything very messy and complicated. And so this was my own attempt at getting through some of it. So why the Krebs cycle brings the Earth to life and our own lives to an end, this is, I know, a little bit posturing. But I have, as it happens, 2 labs at the moment. And one of them is working on the origin of life. And the other one's working on mitochondria and aspects of aging and so on. And it turns out, I wouldn't have really expected this, but it turns out that both labs are trying to measure Krebs cycle intermediates for half their existence. And you kind of think, well, there's got to be something in common here. Why is it so central to everything? So this is my very limited attempt at capturing the essence of what's going on with the Krebs cycle. This is a kind of a, well, this is a bioblast. As Erich said, this is not a mitochondria, and you can tell that. Neither is it really a cell. It's simply it's a conception of the topology. And so we have glycolysis going into the Krebs cycle represented with CO2 and 2H coming out. The 2H is important. I'm deliberately not saying NADH, and I'm showing the respiratory chain and so on. So I've tried to capture the essence and proton pumping across that membrane. But we all know as well that the Krebs cycle is central to a lot of biosynthesis, and it becomes very messy very quickly. And again, I'm capturing this in the simplest possible way without trying to over-complexify it. And it already is quite complex, and you can begin to understand why textbooks would separate this chapter off from the last chapter and really not overemphasize the biosynthetic aspects. What I would say has really happened in cancer over the last decade or so is, you can't generalize about cancer, and I'm doing it anyway, and I shouldn't, but in essence, the Warburg effect is really, do mitochondria need all that ATP, or is it really about biosynthesis? Is it really about, they require membrane potential and so on, but it's really about driving more cancer cells, all the biosynthetic intermediates. So how do we reconcile these two things? There's this yin and yang, as I see it, to the Krebs cycle. It's required, obviously, for respiration and generating NADH. It's also generating the carbon skeletons for biosynthesis and growth. And so it underpins really all aspects of cellular fitness and organismal fitness. And again, it's difficult. We all know this, and yet somehow, when we talk about mitochondria, we talk about energy, and we tend not to think about biosynthesis primarily. Although we know it, I think we underestimate its importance. And so a deficit in respiration necessarily shifts biosynthesis, and that necessarily shifts the epigenetic state of cells. And I'm not sure, I mean, I'm beginning to try and understand what's going on here. Some people do, but a lot of people tend to just think about the energetics. So why is this same cycle used to create and destroy? And I'm linking it now back to the origin of life and the end of life. So this is what came as a surprise to me some years ago now, chlorobium. This goes actually back to 1966, the discovery that some green sulfur bacteria, and actually plenty of other things, and maybe all bacteria, we're not absolutely sure just how widely dispersed this is, run a reverse Krebs cycle. So they're doing photosynthesis, not using the Calvin-Benson cycle as plants and algae do, but they're using the reverse Krebs cycle, and this is a simple conception of it. That's just giving you a sense of how old this is. This is Mike Evans, who just before he left for Berkeley, he left in 1965 for Berkeley. Apparently his car dates back to the 1950s. It was a vintage at the time, so already that's a slightly misleading picture, but it was quite a long time ago. And here it is. In that original 1966 paper, they made the point that this is effectively fixing 4 times as much CO2 as the Calvin-Benson cycle for a single turn of the cycle. And it is a rather beautiful thing. It's incorporating other elements that are not in the conventional Krebs cycle, acetyl-CoA and pyruvate specifically, and phosphoenol pyruvate and so on. And it's basically you're fixing CO2 and hydrogen and so on, and converting gases from the environment into the carbon skeletons right at the heart of all of biosynthesis. Here was the original figure from that 1966 paper. And it really emphasizes right in the middle, reduced ferredoxin. I mean, it's beautiful. You're talking about art, and what are you going to see in that? Well, you're seeing the CO2 going in, and you're seeing reduced ferretoxin. That's what's jumping out at you from there. And there's one of the early, this was 1971, the electron density structure of ferredoxin, showing the 4Fe, 4S clusters right there at the heart of ferredoxin. So some of the early crystallography work on these proteins. So both, that paper was 1966, and this paper was also 1966, also on ferredoxin, which had been discovered in about 1961, I think, ferredoxin. And this is one of my all-time favorite papers. This is from Margaret Dayhoff, Richard Eck and Margaret Dayhoff. And she's been described as the mother and father of bioinformatics. She really pioneered the methods that everybody is using in phylogenetics these days. They came they came largely from Margaret Dayhoff. And this was one of her early studies. It was on ferredoxin, and she recognized that there was a repeating pattern in ferredoxin that would break down to essentially a repeating pattern of 4 amino acids. And I have to read this out to you because she says, the incorporation of the prototype, this ferredoxin, into very early metabolism during very early biochemical evolution, even before complex proteins and the complete modern genetic code existed. She's talking about proteins made up of small building blocks before the genetic code existed. This is really exciting stuff. And it's still, it's still kind of percolating through. And it's percolated through to me, finally, 50 years later, because we're working on these questions to do with the origin of life. So let me just orientate you to that. The most ancient cells that we know about from phylogenetics, things like methanogens and acetogens, they are growing from hydrogen and CO2, and therefore already it's beginning to look like reverse Krebs cycle. In fact, they're using a slightly different pathway, the acetyl-CoA pathway, which runs into the reverse Krebs cycle. They depend on ferredoxin, and they depend on the proton motive force. Occasionally a sodium motive force, but mostly they were depending on a proton motive force to drive their growth. And the way that methanogens are doing it is they're using this energy-converting hydrogenase, the ECH sitting up there in the membrane. And that's driving the most difficult step. And that most difficult step, the problem with hydrogen and CO2, this is still amazing to me. Hydrogen and CO2, mix them together in a flask, warm them to 50 degrees centigrade, put a catalyst in if you want, shake them up. What should happen? What should happen thermodynamically is you should get cells. Cells are more stable than a mixture of hydrogen and CO2. I still find that gobsmacking in the strict absence of oxygen. But of course it doesn't happen. You can shake your hydrogen and your CO2 and you're going to end up with hydrogen and CO2. Why? There is a kinetic barrier, and that's the enzyme that's breaking down the kinetic barrier using the proton motive force to do so. And so this is some work from Rolf Tauer, but under normal conditions, the E prime of hydrogen is about minus 300 millivolts. That's a 10 pascals partial pressure of hydrogen. And ferredoxin, 99% reduced, it's about minus 500 millivolts. So there's a 200 millivolt gap there that the proton motive force is driving that reaction. How is it doing it? Here was my attempt to kind of figure out a process. If protons are involved in this reaction, then it's going to, just through the Nernst equation, affect the reduction potential by 60 millivolts. But you look at channels like this and ask, what does pH mean? It has no meaning in a channel. There's either a proton there or it isn't there. pH or temperature, all these things are bulk properties. And we're dealing with, this is more like Schrödinger's cat. Either there's a proton there or there isn't a proton there. And so, in effect, the local pH is either 14 or it's 0. You've got a switch here. And anything passing across a membrane has really can switch that over. So this goes back to Mike Russell's ideas and deep sea hydrothermal vents, Lost City, for example. Just abstracting this down to the principles, there are high concentrations of hydrogen bubbling out of the ground in these hydrothermal fluids. In the early oceans, lots of CO2, probably 1 to 10 bar CO2 pressure around 4 billion years ago. And there are proton gradients across these catalytic iron-sulfur walls. And the pores in their topology are very similar to, let's call them bioblasts, shall we? That's the topology of a bioblast, as I showed you earlier on. Protons outside, relatively alkaline inside. Same with a pore in a hydrothermal vent. So this is my imagination. We start off with a pore. We have a proton gradient across an iron sulfide barrier. And it is driving through these proton transfer and electron transfer and so on. Essentially the fixation of CO2 by hydrogen to form Krebs cycle intermediates from which other things such as lipids and amino acids are produced. And the entire system is kept running by the through flow of alkaline hydrothermal fluids and the percolation of acid waters. What would be the end point of that if that were really true? Well, the lipids would go, fatty acids would go to form a bilayer membrane, and you get a protocell in this space. And amino acids, well, they'll interact with things, including, for example, iron-sulfur clusters. I'm showing you iron-sulfur mineral there. They would interact with that. Cysteine would interact with that. Maybe it would make smaller minerals. Who knows what it would do? I'll tell you what it would do in a moment. A whole bunch. This is my attempt to capture it, but there's a lot of predictions in there. If this is a hypothesis, it says that these membranes are going to be stable across quite a wide pH range. It says there will be interactions between iron-sulfur clusters and amino acids. It says that a lot of this metabolism will happen spontaneously under the right conditions and so on. And we can test most of these, and I've spent the last 5, 10 years trying to test most of these. Took a long time. Finally, we got formate in this system. I'm not going to have time to go over the results in much detail. I just want to give you just a sense. pH gradients will drive CO2 fixation across semiconducting iron sulfide barriers. And we've been doing it in microfluidic chips. And you can just see a barrier there. This is laminar flow as we have these two different fluids mixing. And we're getting formate, we're getting acetate. We hope we'll get pyruvate, and we're going into the Krebs cycle. Are we getting methanol and things like that in there? That's just an NMR showing it. This is the core of metabolism that we would hope to be able to capture. And all I really wanted to take away from this, top left-hand side is activated CO2, and in brown throughout is hydrogen. Hydrogen, 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 hydrogen. In green is CO2. This is the core. We're going straight into the pyruvate, oxaloacetate, alpha-ketoglutarate, straight into Krebs cycle intermediates, from which we're making aspartate and glutamate and glutamines. You know, we're into metabolism that everybody knows, from CO2 and hydrogen straight away as potentially prebiotic chemistry. The blue arrows in here are reactions that have been done under plausibly prebiotic conditions, not by us, well, some of it by us, but a bunch of groups around the world trying to do this. So it really does happen. We can make protocells, fatty acids, and fatty alcohols. We'll make, basically that you can see there with cryo-EM, protocells that are stable under a wide range of pH, from pH 12 in hydrothermal vents right the way down to 7 or just less. Salt conditions, magnesium, calcium, and so on. Amazingly, with cysteine alone at pH 9, in the presence of ferric chloride and sodium sulfide, you get 4Fe, 4S clusters just spontaneously forming. And you can see there with UV-vis spectroscopy the characteristic shoulder at 420 nanometers. There's also Mossbauer spectroscopy, shows we're getting different types of clusters, 2Fe, 2S clusters, 4Fe, 4S clusters. And cyclic voltammetry shows that they are redox active. So it's amazing. You can make these things really simply. We're able to make uracil, for example. So the pathway, this is the biochemical pathway in the absence of genes and proteins. We were just scooped on this by another group, but at least we both found the same thing. This is reproducible science. Unfortunately, they got there before we did, but hey. So I'm not going to go through this data, but you're very welcome to ask questions about it later on. But this is following the biochemical pathway, and we're able, in front of the graph at the right-hand side there shows that we get about 8 times as much under the set of optimized conditions, which turn out to be high pressure, mildly alkaline, reasonably warm, 70 degrees centigrade. This is one you might like, ATP. This is amazing. ATP, where you can phosphorylate ADP using acetyl phosphate in water. It works best in water. It works less well at lower water activities. And other carbonyl phosphate works a little bit, but in the presence of ferric iron as a catalyst, and only ferric iron worked, we get about a 20% yield of ATP. But at the right-hand side, you can see if you do that with ADP, you get ATP. But if you do it with IDP or GDP and so on, you don't get anything at all, and same with pyrimidine. So why is ATP the universal energy currency? Part of it is it can be made spontaneously in water just by substrate-level phosphorylation from acetyl phosphate, and with no enzymes, with nothing there at all. So all of this makes me think that this is feasible. This kind of thing is worth pursuing. It may not be right, may be wrong in all kinds of unexpected ways, but I'm just going to move on. What happens next? We've got these protocells. There are patterns in the genetic code that suggest that there are direct interactions between amino acids and bases in RNA, for example. They're quite elusive, these patterns. But if we just take them to exist, and we're looking for them in the lab as well, if you take them to exist, what it says is you've just introduced a random string of RNA into growing protocells, and you can template a non-random peptide onto it in some way, then you're introducing information into biology, because a random RNA will produce a non-random peptide. That non-random peptide will have some function in growing protocells. They'll facilitate their growth or interfere with their growth. And so there is no problem with the origin of information in biology if we start with metabolism. Now, for the last 10 minutes or so, I hope I have 10 minutes. Yes? Good. Okay. On the left-hand side is the reverse Krebs cycle as I've been talking about it. We have hydrogen and CO2 producing organic matter. I'm showing this as methanogenesis, producing methane as a waste product. On the right-hand side, what you saw already, the conventional Krebs cycle that we all know, feeding 2H as NADH into the respiratory chain. In the book, there is a chapter about how on earth you go from one to the other. The reality is there's hardly ever a complete Krebs cycle. It's really more of a mad roundabout system with things, traffic entering and leaving at pretty much every exit. And so there isn't really such a problem. But there are some issues. So you can imagine, at least, that if you have a problem with respiration, if you have a problem at complex 1, as I'm showing here, it's not only interfering with ATP synthesis, it's also going to interfere with Krebs cycle flux. You can't oxidize that NADH nearly as well in the mitochondrial matrix if you've got a problem, a specific problem with complex 1. So it should have an impact on all flux through the Krebs cycle. And that is because the Krebs cycle is the basis for the rest of metabolism, then you've got a serious problem with all the metabolism. And we do have some results in flies, which I'm just showing this one result from, where we used an antioxidant, thinking it would work as an antioxidant and be a helpful thing, N-acetylcysteine, NAC. And we found that this is 3 different ways of looking at complex 1, but the red ones here, these are flies with mitonuclear mismatches, so they all have a genetically identical nuclear background, but they've got slightly different mitochondrial DNAs, just a few SNPs different. The red ones have 9 SNPs different to the wild type. And when you give them NAC, their complex 1 linked respiration is suppressed quite dramatically. It's halved, more or less. And you can see at the right-hand side over there that the red ones down at the bottom, that's survival. This was day 10 survival, day 12 survival. So almost all of them were dying. And they seem to be suppressed in respiration at complex 1. We looked into that a little bit, and what you see at the top right is the gray bars. This is ROS flux with an O2K, and it's not changed. And just above that, stacked above that, is with rotenone, and that really hasn't changed either. And it's antimycin A above that. But it's really striking that there's a huge suppression of complex 1 activity, but ROS flux has not changed under the same conditions. And because you can measure them simultaneously, we can be fairly sure, I think, that we are measuring something meaningful there. And the fact that it rises with rotenone is what you'd expect to see, and so on. What does this mean? To me, it's suggesting something that I didn't used to think was the case, but it's suggesting that there is a physiological requirement to keep ROS flux within strict limits, and one of the ways to do that is to suppress respiration at complex 1, to the point of death, by the look of it, in these flies. But there are other ways out of this. There are plenty of other, as Erich has really emphasized for many years, the idea of a respiratory chain with electrons entering at complex 1 and just going straight down, rather than there being a Q junction and there being lots of inputs to complex 3. You don't need complex 1 for respiration, for membrane potential, for driving a lot of things, but you do need it for oxidizing NADH. It's very important for oxidizing NADH, and if you're not oxidizing NADH, then you're potentially going to have reverse flux through parts of the Krebs cycle, not necessarily the whole Krebs cycle. Here we've got plenty of input from glycerol phosphate through glycerol phosphate dehydrogenase, for example. There's plenty of ways you can do it through proline dehydrogenase. Plenty of ways to maintain a membrane potential and generate ATP, but none of them are going to fix your NADH problem in the Krebs cycle. And this is probably, as I said earlier on, this is probably the basis for the Warburg effect. That parts, at least, of the Krebs cycle are likely to be running backwards in cancer, and that's a beneficial thing because it means that it's driving biosynthesis. It means that you are converting, effectively, Krebs cycle intermediates into the basis for making new sugars, new lipids, and so on. But, not all cells can do that. Cancer cells are very good at it. If the entire body tried to get away with doing that, then it would be a disaster, and certainly we'd die. And some cell lines cannot do that because they are stuck. They really do need to use glucose as a fuel, for example. So in insulin secretion, a lot of you in this room will know much more about this than I do, but my understanding of this is that the way in which glucose levels are detected is by fluctuations in membrane potential in the beta cells, which I think is fantastic. And so you have increased glucose, and so you have an increased membrane potential, and then you have a problem that if you've got damage at complex 1, you're not able to respond in the normal way. You don't detect the glucose changes in the same way, so you don't put out insulin in the same way, and so on. So those cells are stuck using glucose and using complex 1, and they're doing it in a particular way because they're using it to calibrate levels. Same, I would say, with a lot of neurons, which love glucose. Lactate's essentially the same thing as an introduction to entry point into the Krebs cycle. So oxygen's introduced, neurons fire up, the pyruvate dehydrogenase complex is activated by calcium, off you go, generate a membrane potential. What happens, though, if you've got damage to complex 1? You've got a serious problem. You're no longer able to respond in the normal way well, you would be less able to think. What do you do instead? Well, I like this work from Estela Arriogomez and Erich Schoen suggesting that, well, you ramp up calcium, and that is damaging to the mitochondria-associated membranes, which are associated with a lot of the other processing of proteins and lipids that are going on in Alzheimer's disease. So I find this a very appealing idea, and again, I'd like to have your take on it. But this is a problem that you have if you're specifically going to say, I'm going to use glucose. I'm not going to use fatty acids as a fuel in a neuron. I'm not going to use amino acids as a fuel in a neuron. I'm going to use glucose, and why? Why do they want to use glucose and nothing else? So for the last 2 or 3 minutes, I just want to say, maybe it's because of consciousness. I'm not sure that Erich is going to agree with this because we talked about this last night, but I'm going to put it out to you anyway, and we can take a vote on it later on. Science is not democratic, of course. So this is what Luca Turin, I don't know how many people know Luca Turin, but he is something of a mad genius. And he has a knack of capturing the simplicity in things. He says almost the only thing we know for sure about consciousness is that it is, so to speak, soluble in ether, chloroform, and other solvents. We can anesthetize people. We can dissolve consciousness. And there's a wide range of agents that will act as general anesthetics on all animals, he says, from protozoa to man. I like the way that he includes protozoa as an animal, suggesting that there's a basic cellular mechanism is involved. And again, I like the idea of basic cellular mechanisms. How can anything evolve if it doesn't evolve in single cells? In bioblasts first. So all kinds of different structure general anesthetics have. There is no single lock and key mechanism that explains the way that they all work. Biological activity unrelated to structure is the way that Luca Turin puts it. And the one that's really most interesting is xenon, an inert gas. It is a general anesthetic. How on earth can inert gas, as he says, it doesn't have any chemistry, it's an inert gas. It doesn't really have a shape. It's a sphere of electron density. But it does have physics, and the physics it can do, and I have no idea how it does this, but it is able to facilitate electron transfer somehow. And we have xenon lamps and so on, which illustrate that. He imagines that it sits in hydrophobic pockets in proteins and somehow facilitates electron transfer, and has demonstrated using EPR that there is an oxygen signal, that anesthetics affect the oxygen signal. Now, you would say that that's okay, of course, you put someone under a general anesthetic, it's going to affect respiratory rate, it's going to affect oxygen consumption, but if you're using xenon, then what else can it possibly be doing? Which means this is still a correlation, this is not cause and effect. However, it looks much more persuasive that there is something directly related to a transfer of electrons, short-circuiting the respiratory chain, must have an effect on membrane potential. But it's very, very, very difficult to measure membrane potential, so how on earth would we know? And the reason I say that is because what we're really measuring when we're measuring membrane potential using Nernstein dyes that accumulate in the matrix is we're measuring the difference between the matrix and the cytosol. We're not measuring the difference between the matrix and the closed Christie space, which is closed, and those protons really are in that space. And there's practically no water in there. It's all structured water in there. It's very, very difficult to know how can we actually measure. There are some very clever ways of possibly getting at it, but I don't really know if they work. It's difficult to calibrate anything. But the other thing that we've learned over the last perhaps 5 to 10 years or so, I'm showing ATPase dimers at the end of the Christie here. The Christie structure is structured by the proteins that are sitting in it. And the ATPase dimers are sitting right there at the end of the Christie. My understanding is the pumping complexes are generally in the walls of these, and what that means is we have a proton circuit. We have an oscillating electrical charge, and that means mitochondria should be generating electromagnetic fields. And those electromagnetic fields will interact with each other, and if we have multiple parallel cristae, they can either be amplified or they can be quashed. And we don't know, I don't think we know, what the EEG actually is. Of course, we know we're measuring changes in voltage, but of what? We know it's in neurons, networks of neurons, but is it really depolarization of the plasma membrane, or could mitochondria be playing a large role in what's actually happening there? I'd like to think they could be playing a large role. So why, then, if that were true, why would the bioblast be controlling our conscious state? What is it? Why would a bit of the cell be responsible for a sense of the stream of consciousness? Well, that's because they are bioblasts. They were cells once upon a time. I've gone back to my original demonstration of a cell with a charge on the membrane. That membrane is the difference between the entity that is the cell, the self, if you like, and the outside world. And the membrane is, as Peter Mitchell himself said, it's the interface that is separating and connecting the inside of the cell from the outside world. And we have substrates, we have oxygen, we have all of these things. How do you integrate them all into a single system? It's not, I don't think, through gene expression, really. This is happening in nanoseconds or femtoseconds or something. How do you integrate a whole cell as a unit? Well, you certainly do it by having a proton motive force across that entire membrane, and you certainly would be generating electromagnetic fields in that case, and those electromagnetic fields across distances of cell-like distances would be training the molecules within the cell into certain oscillation states, for example. I'm hand-waving now because I don't know. But we see something like the Krebs cycle, which is integrating the environmental status and the internal status with the membrane potential, which is giving you a moment-by-moment feedback on your status in relation to the world. Does that feel like anything? If bacteria die, if they kill themselves, and they do it by depolarizing the membrane, they just effectively pull the plug and allow the protons to come rushing in, that will kill them. It happens a lot with viral infections and so on. The first thing, if you don't want a virus to proliferate and take out the entire population, then apoptosis arises in bacterial colonies, and the point is you can prevent the viral spread. Does that feel like anything? Surely it must feel like something to a bacteria that suddenly depolarizes its membrane. So I'm going to finish with this note that we have the origins of consciousness, not as we know consciousness, but as some kind of stream of relationship with the environment that integrates the cell as a unit, is all linked into exactly this same system. All the data I've talked about, I'm not going to accuse anyone in my group of sharing some of my madder ideas, but all the really good data that I showed you came from quite a lot of people now who've done some fantastic, some beautiful work. And I'm very grateful to them to being associated with me at all, this madman, and being brave enough to address some of these questions. And I guess I have time for questions. I hope I do.
Yeah, we're a little bit ahead of time. Thank you for staying on time. So if you have a question, please remember to raise your hand in your seat, and when we pick you, please say your question nice and loud. Adam.
Hey, thanks for, as usual, very inspiring. Oh, sorry. Thanks for the inspiring talk. Can everybody hear me? Okay.
Yeah.
And I want to stay in the context of living, cells with this question. I love the origin of life ideas and all these ideas that you put out here. Fascinating. But I'd like to get real quick, if I could, and maybe this will have to extend beyond the Q&A, but your thoughts on coupling. That is, the coupling between ATP synthesis and respiration. As you know, biosynthetic processes involving the TCA intermediates that can be generated by the conventional forward reactions are also, used
Sure.
in cancer cells. And part of this Warburg effect is that perhaps the ATP would be coming from glycolysis, but then we still need respiration or all these redox reactions that the mitochondria generate in order to generate these.
Yeah.
But you're limited to some extent by the NADH to NAD ratio, which is limited by the ATP to ADP ratio, so essentially energy demands. So if you wanted to make a lot of things, but you didn't need a ton of ATP, the solution to me would be to uncouple respiration with
Yes,
synthesis.
and plants do that And plants, I mean, the penny dropped for me in reading one of Lee Sweetlove's papers about non-cyclic flux through the Krebs cycle, and he was coming from the point of view, as any botanist would, that a lot of ATP synthesis is happening in the chloroplasts. And yet mitochondrial function is required. They will often, they have the alternative oxidase, they often uncouple as well. It's obviously under extreme control, but they really don't need their mitochondria for much ATP synthesis. They're using it for biosynthesis as well, and for redox balance in general. And so then you think, okay, so are we doing this same thing as well? And the answer is almost certainly not. Why not? Well, because we probably have a much higher ATP requirements generally, and so we're less free to do that. And secondly, if you uncouple, you generate heat. And if you're generating bucket loads of heat, then you're going to have all kinds of other problems as well. So lots of simple metazoans, basal metazoans I would call them in a hand-waving way, do have an alternative oxidase and can do exactly that. They're effectively, but they're small and they have massive heat transfer, and so they can do it. So I think we are big and we have a problem with it, and we get around those problems in fiddly ways. And I have a feeling it's a lot to do with aging because there's only a limited number of ways of doing it. Now, I think if you have really good complex 1 function, you're able to do really good biosynthesis and really good ATP synthesis at the same time. And we've got some data that if you have a look at Enrique's poster, complex 1 function is associated in flies at least with high fertility, good longevity, they live longer, these Barcelona flies, you know, we can see that they've got slightly increased complex 1 linked respiration. And they're basically fitter in every respect. They're more active, they produce more offspring, they live longer. I mean, you think, well, yeah, I could do with some of that. So what are you going to do? You better keep your complex 1 active. And I'm back round to saying, I'm going to go for a run and I'm going to try and eat a bit more salad.
Excellent. Just a quick follow-up. I don't think
Yeah.
it's there. What about fine-tuning? You know, you're uncoupling proteins. UCP1, of course, we have in brown adipose tissue. And of course, there's 5 or 6 others. Do you think they play any role? Obviously, not in switching from fully uncoupled to coupled, or vice versa, but in any real relevant way, modulating this.
I'm sure they do, and I'm sure it varies from tissue to tissue. And again, one thing that came up discussing with Erich last night is. Erich's very skeptical of this idea that the mitochondria would be generating electromagnetic fields because you would require a circuit, and those circuits would have to be parallel in different mitochondria. It would really have to be very well organized for this to work, and he's quite right about that. But the other thing is a membrane potential in itself is not going to generate an electromagnetic field. It's the cyclic flux that could do that. Now, that means that if suddenly a neuron is booting itself up, and we've increased pyruvate dehydrogenase, allowed calcium in, and so on. Then suddenly we've ramped up not just the membrane potential, but electron flow and proton pumping. So we would, for a short period, have, I suspect, it was difficult to detect some kind of sudden ramping up of fields and things as well. I suspect that a lot of the UCPs and so on are linked with an association between flux in space, between oscillating charges and the membrane potential itself. And I think there's probably a whole series of regulatory mechanisms there that we've barely begun to look at because it's so difficult to measure them.
One more question for 1 minute.
Very brief. In your evolutionary perspectives, you emphasize that the evolution of higher forms of life need mitochondria, oxygen because of ATP, because of energy. Now, how do you reconcile this with your now downplaying the ATP requirement even for growth in cancer cells?
I'm not really downplaying it so much as upplaying other aspects of it.
Okay, that is the message, I think.
Okay.
All right, thank you very much, Nick.
Thank you.
Our next speaker for this session is Dr. Lisa Chakrabarti. Her talk is entitled Going South, an Antarctic Expedition to Understand More About Mitochondrial Hemoglobin and Aging.
Wow, it's very nice to be here. It's really nice to see all of you here, and thank you very much to the organizers and to Ouroboros for asking me to tell you a little bit about what I've been up to most recently. So going south is also a term used for getting older or things not going quite as well as you'd want them to anymore. And this is related to my research subject, which is mitochondrial aging. So this is me standing on the deck of Polar Stern, I think in April this year. So I think it was Easter Sunday, where we had this beautiful weather. And I was in the middle of the Weddell Sea in Antarctica. I'd like to tell you why I was there. Let's see if I can move this along. So Polar Stern is the research vessel. It's the flag, literally the flagship of the German government research vessel. And Polar Stern is an icebreaker that can go into the Antarctic and recently spent a year in the Arctic, floating through the Arctic ice. It's a very impressive ship. So let's see if I can get this technology right. Hang on. Here we go. So aging. Nick introduced a little bit about aging. Aging is what I'm interested in. And aging happens as a function of time, I think. Some people will say it's a disease. I don't tend to agree. But aging happens, time goes on, and people get older. People even get older in Innsbruck in Austria. And one aspect of aging that we're all very interested in or familiar with is the mitochondrial aspects of aging. And whatever we do, this mitochondrial aging changes us, either for the better or in some cases, in some aspects for the worse. So where did we start? So I started my career mostly looking at the brain, looking at neurons, then looking at neurodegeneration. But you can look at disease all you like. Until you know what's going on in the healthy organism, it's really difficult to understand. So one of the earlier experiments that we did with mitochondria was to take the lipid fraction from mitochondria from mice, from their brains and from their skeletal muscle, and we took mitochondrial fractions from young and old brain and skeletal muscle. And we did a lipidomics experiment where we were able to show quite clearly that mitochondria are different depending on which tissue you get them from. And they also are different, but predictably different, so that you can do unsupervised PCA, and they will fall out into separate groups as to what, according to whether they're young or old. And so this was work done by Amelia, a graduate student in my lab, some time ago. And this got us thinking about, so if we start looking at healthy mitochondria and look at the lipids or the proteins that change in them, then maybe we can get a handle on various diseases. Let's see. You can use the presenter. It's easier. Okay, so I came back to neurodegeneration, and we had a mouse model of neurodegeneration called the Purkinje cell degeneration mouse, and this mouse undergoes cerebellar and retinal degeneration in a very predictable manner. And we did some proteomic work on mitochondrial fractions to follow up the lipid work. And to our surprise, we found that hemoglobin showed up. And at first we dismissed this, and then we thought, actually, let's follow this up. Actually, there was a very insistent undergraduate student of mine, Oliver Haygate, who said, no, this is real, you have to look at it. So we did some immuno-EM and various other subfractionation studies, and were able to show that hemoglobin is in the mitochondria. Also, we were funded by the Michael J. Fox Foundation, and we were able to show that in Parkinson's disease brain mitochondria, there are changes or there are differences in mitochondrial hemoglobin content. We also found that if we looked at, in fruit flies and put them through various cycles of hypoxia and normoxia, that we could modulate the levels of hemoglobin in the mitochondria. So, normally, when you find that hemoglobin or a protein is changed and you're interested in following it up, you make a knockout mouse, or you would make a knockout mouse. Hemoglobin is very difficult to do that because you're obviously not going to end up with any kind of animal at all. And in my search search around hemoglobin, I came across these ice fish. Now, ice fish are a family within the suborder Notothonioidea that lack hemoglobin. So these are the only vertebrates that don't have any functional hemoglobin genes, and they also don't have any red blood cells. Some of them also lack myoglobin. And I thought, well, how can these possibly exist? But they do. And there's a whole bunch of people looking at ice fish in the Antarctic. The first thing we did was we contacted people. So this is Chiara on the right and Magnus on the left. And we're standing in the University of Padova in front of Galileo's lectern. And Chiara and Magnus have been looking at fish in Antarctic waters for some time. And we were able to get some tissues from these fish. And with those tissues, we were able to do some mitochondrial proteomics, and we analyzed pathways, networks, and various different interactions using co-immunoprecipitation methods. And we had our first paper out looking at these fish. And so this paper was authored by Gunjan and Brad, who's presenting a poster on fly. Our fly work is also in the audience back there. So after a bit, when you've got an O2K in your lab and you're looking at mitochondria and you've got very interesting mitochondria, you think, I really want these live tissues so that I can do some respirometry on them. But the only way to do that is actually to go to Antarctica. So I was lucky enough to hitch a ride on Polar Stern. And if you see on the top right there, there's a map showing how we sailed from Cape Town to, now this is going to be a problem. How do I start the videos? If you can start them for me, maybe. Yeah. So we journeyed from Cape Town through the Weddell Sea. Oh, sorry, that's the noise. That's the sound of the ship. And then up to Chile. And we traveled over 2 months through the Weddell Sea to collect samples. And this is what it's like fishing for ice fish in Antarctica. It's very cold. It's icy, it's snowy. This is Sarah and Neil's waiting for a catch to come in. There's a lot of waiting, there's a lot of being cold. You can see here there are icebergs right next to the ship, and on the working deck you could probably very easily just fall straight into the water if you weren't careful. So more pictures, because now we're not really talking about science, we're talking about Antarctica. We were fishing in the middle of the night. It was scary. We had steel toe boots on, hard hats on, and then when we got the fish, it could be 2 a.m., or when we got the samples, there was a lot of sorting to be done. So this was, the whole trip was to set up an Eastern Weddell Sea observation system where we went through all the different samples that we caught and monitor and set a baseline for what's happening in the Antarctic seas at the moment. And on top of that, we're interested in climate change. So the ice fish work that I was doing on board was to do respirometry at different temperatures on the ice fish mitochondria so that we could see what the potential effect of climate change would be. So a quick rundown of the various creatures we saw. So that really cute little thing is a microscopic, very baby sea cucumber. And then we had ice fish the size, well, that weighed about the same as me. So, you know, very large, no, this isn't an ice fish, toothfish, that are very large. They're the Chilean sea bass, if you've ever eaten those. We had shrimps and krill, anemones, sponges, sea stars, all different sizes, and then there were people that were monitoring the ocean floor and taking images. Here you see a sea sponge with crinoids decorating it, and of course our lovely fish. So the I'm failing with the pointer here. No, here we go. So this is the ice fish. This is one of the first live ice fish that we were able to land. It's a Chionobethyscus devitti. Really hard to identify the specific fish. I'm running out of time. We saw lots of whales and seals and penguins, and of course we did lots of respirometry, and we had lots and lots of weather. Videos? So we had lots and lots of weather, lots of storms, and icebergs. lots and lots and lots of different icebergs. So I just want to thank the whole team, all the crew on Polar Stern, and I'm finished. Thank you very much. Ask me questions if you want.
So we're a little bit ahead of schedule, so maybe a question or two?
Thanks, Lisa. Just real quick, the hemoglobin you're seeing in the mitochondrial membrane space, is this the full out, 64-ish kilodalton hemoglobin we see in red cells, or is it a half, more like a myoglobin? It's the full protein?
We think it's the full protein. We don't have really conclusive data to show that yet, but our hunch is that it's the full protein. Yeah.
And do you have a sense of the respiratory optima then in different temperatures with these ice fish?
Yeah, it's actually very interesting. So they seem to be able to compensate for a certain number of degrees. So they actually exist at around 0 degrees. And so they can compensate for several degrees rise in temperature, and then everything just bursts, disappears, doesn't work anymore. Another interesting feature to note, though, and this is something that we don't talk about very much, is the idea of pressure being an important feature as well. So these fish are benthic organisms. They live very, very deep in the sea. And so what kinds of, forces those are exerting on the system?
Offset the temperature in some way?
Yeah, well, so you would think that there would be some kind of relationship there, but that's a difficult one to test.
Well, isn't it mainly the high lipid content that supports oxygen diffusion, and then particularly the myoglobin is not required because oxygen is so highly soluble in the lipid phase and at low temperature also?
Certainly that's one theory.
That is Bruce Seidel's approach to explain the pattern.
It's entirely possible. These fish have a lot of mitochondria as well. And they have other features. They have these antifreeze proteins in them. There are lots of different ideas about exactly how these fish manage their oxygen supply. And I don't think we really know yet. I don't think anyone's got super close, but yeah, definitely lipids are I like lipids, as you know.
Other questions? All right, not seeing any. Thank you.
Thank you very much.
And that takes us into our coffee break. Yeah, it's coffee break time.